colleague of organization, and our most wonderful volunteer, Wout de Jong, went last year together with me to the Congress in Denmark, and there were 120 people, 1.5 days, talking about the health, the taking care of, and the wonderful hobby of koi. So we came to the wonderful idea and said, okay, next year we do the Holland. I think there's a lot of interest for this type of Congress, but maybe we have to work a little bit better out next year to get more people to come to this event.
For inviting me. My name is G.Wiegertjes. I come from Wageningen University. Everybody's trying to take me. Does that mean I can't walk around? Or will you follow me when I walk around? Because I like
you can walk.
Okay. Can you hear me at the back?
Yeah, okay.
Good. I was asked to tell you something about the immune system of fish. Right? I see a familiar face. I have no clue how much you know about the immune system of fish. So before I start, I would like to quickly test what you know. So Influenza, she works with me and she's sitting here and she will give the presentation. After my presentation, we decided to split it up. That is because it's much more interesting to listen to two different persons for 2 times 45 minutes than to one person for 1.5 hours. Because we study the immune system. So that means we'd like to study an immune response to something that we give to the fish and not something that is already in those fish because they come from a pond and they're harboring bacteria, viruses or parasites. No, we'd like to have them as clean as possible. We'd like to call that specific pathogen free. And then when we give this fish a virus or a parasite, we know that we gave it. Is an evolutionary tree of the invertebrates, so of all invertebrates. And what we found out is that invertebrates have innate immunity, so an inborn immunity, but no acquired immunity. And acquired immunity, I mean, most of you say B cells, T cells, yes, I know about this. Acquired immunity is the most important thing if you want to vaccinate an animal. So those animals or those invertebrate species have only innate immunity, and we're finding out. I won't tell you a lot about that today, but in my lab we work a lot with a particular parasite. It's a trypanosome. It's that if we know more about the acquired immune system of a carp, we can try to vaccinate these fish. Then the other reason we study the immune system, of course, is practice. We have a large culture of carp. I've put a picture of zebrafish and a picture. Do you also recognize this one? Yeah. So why did I put the pufferfish on? Anybody has a clue? Anybody else has a clue why I think that's an interesting fish, apart from its looks? For us, this is very important because they've unraveled the genome of this fish. And the zebrafish there's maybe a very small one. Number of immune organs that are very important, like the thymus. The thymus gives you your T cells. That's where the name is derived from. You have bone marrow. Bone marrow gives you the B lymphocytes. The name is maybe derived from the bursa, from fabrices, from chickens. But the B, bone marrow, is easy to remember. So and we say, look, a granulocyte, a neutrophilic granulocyte, if you look through the microscope, it should have a lobed nucleus. If you go to a human, have natural killer cells. And then I have to be careful because I say fish. We know that with quite some certainty from catfish, because there's a group in the United States that has been doing a lot of studies on channel catfish, and they have identified those cells. To understand, because immunostimulation or immunomodulation is a hot topic, and to my opinion, it is based a lot on these type of receptors. So these are what we call toll-like receptors. You get a signal inside the cell that the cell has to do something. It has to send out a signal to the rest of the immune system. There is something happening, there are pathogens we don't want to have. Side, TLR5 recognizes flagellin from bacteria that have flagella. Two concepts, humoral and cellular immunity, cell-mediated immunity, are two terms that have been used a lot in the immune system. But that's a totally different concept from cell-mediated immunity based on virus-infected cells. So when a cell is infected with a virus, you don't phagocyte. Very good in agglutinating antibodies. You can see it can easily form clumps. And when it can form clumps, it can do different things for the immune system than when you would have only this one. So immunoglobulin G, this is not a knee. This is a T cell and an antigen presenting cell. So an antigen presenting cell is just another name, let's say, for a macrophage. So your macrophage is a phagocytosing cell. It phagocytoses your microbes. It digests them and it cuts them in small pieces. Have the immune response. So this is the number of days after you first see your microbe. Here your B cells, your B lymphocytes are naive. They've never seen this microbe before. Your T lymphocytes are naive. They've never seen it before. Then they recognize something. The T lymphocytes recognize your microbe taken up by your macrophage and digested and presented by the LHC. The B lymphocytes may recognize it with the immunoglobulin, the antibodies on their surface. So what I've just told you, the immune response, you have the immune response going up, and then you have your cells dying off. Those were still a matter of days, maybe up to 2 weeks. Now we're talking weeks. So here's a scale of 12 weeks. So you start again with your naive B cells, and you get a primary response. Your B cells start to be activated, you have a lot of them, you have a peak, you get an antibody response that goes up, and then you get your apoptosis, and your cells die, but you keep the memory cells. Those are handy, and that's the basis of your vaccination. Yeah? And then of course you get a declining phase at the end, but this, you see here, this remains high, and quite high. So your antibody response stays high for a long period. What I haven't dealt with, what tomorrow you will deal with, for instance, is the stress influences of stress on the immune system. The temperature, of course, I've grown up with working on fish, so to me it is very common that the immune system changes with temperature when you work with fish. So I tend to forget that when I talk to somebody that works with mammalians, or with humans even, that the temperature doesn't change when you go outside and it's cold, or that the immune system doesn't change. So to me, the relationship between temperature and immune system is very important, and it has enormous impacts. If you want to vaccinate your fish, if you do it at 6 degrees, it's completely different from when you do it at 25 degrees. Although, of course, you should do it at 25 degrees when you want to vaccinate a salmon fish. So the temperature is an enormously important factor. The rules of vaccination I also haven't dealt with. Maybe it's good to know that a number of years ago, when vaccination in fish started, then people really thought that the bath vaccinations were going to take off, because there were a number of vaccines, a number of bacterial vaccines, antibacterial vaccines, that worked very well when you put them in the water. So you could just take your buckets, you could put your small fish in, the vaccine would be taken up by the gills or the skin, and the fish would be protected. And everybody thought this is going to be very easy. But those were only successful for 2 or 3 vaccines, 2 or 3 bacteria, and for the rest it stopped working. But it's still the way to go for fish. Complicated, I would just like to give this to you. If you bring this back home, you understand the immune system. This is Obelix, you all know Obelix. This is your innate immune system. This is the macrophage. This is the one that comes in first and eats anything it sees. It doesn't differentiate, it just kills what it sees, as long as it's alone. This one is much smaller, it needs help. It needs cytokines, it needs signals to say, hey. But then when it comes in, it goes for the leader, it picks out the specific ones, and it picks out the small ones that are left, after this one cleaned up most of the mess. So this is basically your immune system in a small slide. I think this one you should be able to remember. On the other hand, if it all was far too simple for you, we could always make it more complicated. We do that each year. We do that each year in April. We do it also in April this year, and then we have an international fish immunology workshop where we always invite the fish immunologists from Europe, where they give insight in the latest developments. They do it at a PhD level, but they teach. So it's not they give you 15 minutes of high-tech immunology already, no. They do take you with them from the beginning, and they try to explain to you how it works. But then they spend, instead of 45 minutes on the whole immune system, they spend 45 minutes only on a particular cytokine. So be prepared, but it's very interesting networking. So if you're interested, talk to me. Maybe you can still squeeze in, and otherwise you come next year.
Really surprising what you can learn on that type of congress. So if you are interested to go there, I can recommend it. It's really nice to be there, and sometimes it even is for us who learned a little bit more about it. It can be really hard and sometimes difficult, but at the end of the day your head is smoking about all the information you have. But I must say, I've been there, it was really great. So if you have the possibility to go, please go and try to learn more about it, because there are a lot of nice studies done in the world about all the different fish species, about immunology, about vaccination, and it's always nice information which you can take with you on your further way.